I want to push our discussion into talking about some of the upstream processing considerations, right? And obviously we start with the plasmid. And I would argue with you, Stu, right, too, that I don't think that the plasmid gets as much conversation as it probably should, considering we're still trying to figure out its quality, right, and the impacts and the ramifications it might have on the product quality. So I'm curious, are there plasmid quality attributes today that you found to be most important in producing high quality mRNA? How are you approaching the world of the plasmid? What learnings have you gleaned so far in your work?
Yeah, there's kind of several quality attributes of the plasmid I think are really important. I mean, obviously, like percent linear is important.
Okay.
If you have residual circular plasmid in there, you'll take a hit to your IVT yield. It can end up producing, run-on transcripts. So you're just making, off-target mRNAs, the incorrect sequence. You can make a lot of dsRNA that way too. So having residual circular plasmid is very bad for the IVT. So that's kind of one of the most important. And then hand in hand with that is the integrity of the linear plasmid. If you run on a gel and it's smeary, you're going to make smeary RNA. Your RNA purity is going to be low.
Smeary RNA. Ooh.
You're going to have a lot of fragments that might have that T7 promoter, but don't have your whole, mRNA transcript. So you're just going to make truncated versions. If your encoded poly A tail is missing, that mRNA is never going to express. So, integrity is really big. And then one thing that's kind of a little more interesting, and I guess not as clear, is percent supercoiled. So we know from like the AAV space and other therapeutics that percent supercoiled is big for plasmid transfections and things like that. But I think it's kind of underrated for mRNA.
Interesting.
The open circular form, nicked plasmid, it can cause, more abortive transcripts, cause the polymerase to fall off, reduce your yield. And then also a thing that plasmids love to do, especially plasmids with encoded poly-As, is form multimers and all sorts of weird recombination multimers where the tail is missing on half. I've seen, you get a dimer plasmid and there's only an encoded poly-A on one half. You linearize it and it looks fine after you linearize it. But then half your mRNAs are missing a poly-A tail because the is missing that tail. So there's a lot to go there. And then residuals too, right, are important, like residual RNA and DNA, like host cell RNA and DNA. You don't want that carrying in your IVT. The T7 polymerase is very promiscuous. It'll grab onto anything that looks like a promoter and make RNA. So you definitely don't want any, unwanted DNA or RNA in your IVT. But sorry, I kind of covered almost all the quality attributes of plasmid, but
you're good. No, you're good. So I think you laid it out really beautifully for us. I love the word smeary RNA and promiscuous polymerase. So I feel like there's a title in there somewhere for a future live event. I'm not going to lie. But I want to jump to Marc and Christian, and maybe Marc, we can start with you. But what are some of the biggest questions? I mean, Stu just outlined a bunch of different quality attributes, considerations for us as it relates to plasmid quality that can go on to impact the quality of our products. But what are some of the biggest questions that remain on your end, right, as you're working in the circular RNA space? About how does that quality of the plasmid impact mRNA coming out of IVT?
Well, I think the quality for us with the circular RNA is more of what impact does the quality have on the formation of double-stranded RNA. I think what I'd love to be in a position in the near future is, I'm not even talking about plasmid DNA, I'm talking about synthetic DNA templates. It'd be great to be able to circumvent the need for bacterial fermentation, the lead time, the extra cost. I think we could make a major positive impact on the cost of goods if we could move to synthetic DNA templates.
Absolutely. And I think we're starting to see a lot more of those kind of, coming out onto the market today. Christian, you're over there nodding. Have you explored this at all, or is this a topic of interest to you?
Yeah, that's definitely an interesting topic for us. And as you said, there are several suppliers now providing that material. And of course, it's of interest for us for the reasons mentioned.
Yeah.
But on the other hand, I have to say, plasmids manufacturers are using their technology for quite a while now, right? It's not with the mRNA that plasmid batches became interesting. It was for AAVs, it is for plasmid delivery itself, and was starting material for many processes. So they're also quite established CDMOs available. So many of the topics you have in small scale and in development phases, you don't have to think of them when it comes to GMP grade material. So also we have great experience with the CDMOs we are using, but of course, the fermentation-free process would be beneficial due to several reasons.
Yeah. Are there any, additional questions on your end too about how the quality of the plasmid impacts the RNA that's coming out of IVT? Anything that makes you kind of scratch your head? How did this happen?
No, I don't think so. So at the end, it's quite simple, right? You have the template, and it's run off by the T7 polymerase, and it delivers you an mRNA. Of course, it has to be pure, it has to be high quality. What may be interesting is if there are any, let's put it a different way around, if template stability is a limiting factor in the IVT. I'm not even sure if data is available on that, but right, the double strand opens and closes over and over again. So there could be causes for inefficiencies, but as said, I'm not even sure if there is not even literature on that topic already, but that might be interesting. Apart from that, I think it's quite established.
Okay, that's good to hear.